So our next presenter is Liz Falendysz. She is an attending veterinarian for USDA Wildlife Services. Her topic today is going to be avian enrichment in a rehabilitation or research setting. Liz, go ahead and take it away.
So as was just announced, I work for the USDA Wildlife Services. So I work at the National Wildlife Research Center. I'm the attending veterinarian there, and I'm a relatively recent addition to the center. So I was previously the attending veterinarian for the U.S. Geological Survey's National Wildlife Health Center. So much of my presentation is going to be discussing the birds that we used in research at both centers. And I also want to start out by recognizing all of the animal care technicians who are really the drivers behind a lot of our enrichment programs at the two centers. They're listed alphabetically here for the ones that some of the many technicians, but they really are the driving force behind creating the ideas and implementing a lot of the enrichment. So thank you to them. So just to give you context for some of the research that I'm going to present setting, the USGS works to understand the pathology and transmission of wildlife disease and develop tools to mitigate disease, primarily at a population level. And the Wildlife Research Center has a similar wildlife disease mission, but also a broader mission to reduce human-wildlife conflict. And so we might be, most of our studies are either with wild-caught animals, birds and mammals or reptiles that we'd be bringing into captivity, but we can sometimes use captive-bred animals or even agricultural animals to serve as models for those wildlife species. So to kind of broaden this from just the experiences that I've had over the last decade or so, I did a literature search for how are birds used in research. I just looked for avian research, and the really broad breadth of types of research that birds are used for is really staggering. Because birds have eggs, they have long been used for developmental anatomy and developmental biology research, and they continue to be well used for developmental toxicology research because we do have that egg where we can observe development of embryos. Likewise, their eggs are often used for vaccine production, and we sometimes use birds to develop vaccines for other avian species. They are used in a wide range of cognition and auditory processing. A common model that I'm going to talk about is the zebrafinch, and they're used widely for auditory processing because passerines have these complex songs that they need to learn. So learning and cognition is another common one. Sometimes our birds are used as comparative models for human diseases, such as in oncogenesis, when we're understanding how cancers arise, including gliomas. We can understand neurochemistry, retroviruses, leukemia, many other things that are applicable directly to human health are learned from bird species. And as I described with the discussion of wildlife disease, we also often may be using birds in research to help their own species or another species of bird to understand things like pathogenesis of avian disease, transmission of things like avian influenza. Avian influenza in particular is a very commonly researched topic because, as was just mentioned, birds are the host that transmits the virus and moves it around the environment. But as we know, with the current high-path avian influenza outbreak, it can affect human and domestic animals. And so it's really a global health issue. And lastly, and certainly not least, individual animals are benefited, individual birds, when we understand better how to rehabilitate and treat their diseases. And so some avian research is for us to better understand things like the biomechanics of avian bones so we can fix their fractures, or to understand their drug metabolism so we can appropriately manage their pain with drugs. So Next, I wanted to just talk, these are some of the most common avian models, but all different kinds of avian species are used in research. So in the middle, we have a Japanese quail or Coturnix quail. That's a common research model. Domestic fowl like waterfowl, mallard ducks, and domestic chickens are also very commonly used as research models. The zebra finch has been mentioned previously. It's one of the species that we had in the National Wildlife Health Center. We had a colony of, and it's used very frequently for both infectious disease and neurobiology, neurodevelopment research. And then cytosines are actually commonly used to help us understand how avian species metabolize drugs. And so the UC Davis, for example, has yellow-naped Amazons that they use regularly to help us understand how birds differently metabolize things like pain medications and how we can utilize those to better treat our captive avian species. So before there were very specific avian regulations from the USDA, many research institutions also followed guidelines from the Public Health Services Office of Lab Animal Welfare. And so a lot of the enrichment program that I'm going to present was actually developed utilizing the tenets of the Guide to the Care and Use of Animals, which is created by the Office of Lab Animal Welfare. But they certainly apply just as well to the new USDA regulations. But I really love the definition that the guide uses for the enrichment and what is enrichment for our research animals. So the goal is to enhance animal well-being. That's the why behind what we're doing with enrichment. But the how is we're utilizing sensory and motor stimulation to do that. And I love that they've included expression of species typical behaviors, because that is really the guiding force of how we are designing our research enrichment programs in trying to show and display those species typical behaviors. But it's also really important that we're utilizing our knowledge of the individual species for all types of housing in determining what's best for that species. And we do that, again, more details of the how is through exercise, manipulative activities, and cognitive challenges. And we're all doing that to promote psychological well-being. And another kind of reason that I like to think of specifically for wildlife is as a tool to reduce stress. We are often bringing wild animals into captivity, which creates a lot of stressors for them. And our enrichment can be a tool to help reduce that stress. So some of the benefits of enrichment for research animals, these are just a few of the general categories, are reduced stress, improved cognitive function in those animals, improved learning, and as I mentioned, improved well-being. And I wanted to mention that my job as the attending veterinarian for a research institution is twofold. So it's to advocate for the research animals, so advocating for their health and well-being, but I also really want to support the researchers and help them achieve the really fantastic and necessary science that they're doing. So I also, even though they now have to be doing these enrichment programs, I've long spent a lot of time reminding or convincing researchers why enrichment is so important. And it's not just for the animals, it's really also for their research. So this is a really nice but complicated figure from the publication I listed below. But it describes how stress can really change the animal's physiology, and that is going to make them a very different research subject. So it's describing what's called the HPA axis, the hypothalamic pituitary adrenal axis, which is how the animal's brains process input from their environment and leads to stress. And so most people know that stress can be a negative thing, especially when it is very acute and a strong stress response, or when it's a chronic stressor. So you may be familiar with the terms of adrenaline for that fight and flight response, and cortisol for that long-term chronic stress. And the output of that are changes in behavior, in the autonomic and neuroendocrine systems, and the immunology system, or the immune system. And so if you're studying those particular aspects, chronic stress will affect the research results. And so enrichment can be seen in two ways. And the other thing I loved about this figure is that in the upper right, they mentioned boredom. And so you can, obviously this isn't a bird in the lower right hand corner or lower left corner. It's an elephant, so it's a mammal, but it's an animal that has advanced cognition, that's developed for foraging in really complex environment that requires learning in order to do that in the wild. They have complex social structures, and this particular animal is probably captive bred. And all of those same attributes could easily apply to a citizen parrot that is captive bred, but that utilizes a really complex cognitive behaviors and in order to navigate their natural environment and do the things that they need to survive. And so in this figure, we're seeing that when you have an animal that has advanced cognition and complex social structures, a lack of input, normal input to the brain, which you would call boredom, can contribute directly to stress. And then, as I mentioned, for our wild caught animals, we might also use the same boredom could apply, but also we may use enrichment to reduce the stressors in our environment. As I mentioned, the stressors could be removing the animals from their native environment, the actual capture experience, and putting them in a novel housing situation. We often alter their social structure by removing them from the native social groups that they had and asking them to create new ones, or ones that might not be completely natural. We often have to handle the animals on a regular basis to observe them or collect samples from them. And the animals that are encaged for our research, they may not have the same amount of control of their environment that can be a stressor. So birds, just to acknowledge, birds' HPA axis works a little differently. They have arginine vasotocin as a primary releasing hormone from the hypothalamus in their brain. It occurs in a different part of the brain, and they use corticosterone instead of cortisol. But the effects are essentially the same. So it does change their homeostasis, changing metabolism, immune function, and behavior. So what does corticosterone do for that bird? It decreases both innate and adaptive immune responses. It can alter neurochemical responses and neural development. And if you're going to be studying neurodevelopment or neurochemistry, that is a very, very important thing to control. Because of the fight or flight response from that acute stressor, that will alter metabolism and GI function, that can alter circulation. And as many of you who've ever had to sit in a lecture and trying to learn something while you have a lot of external life stressors, stress decreases learning ability. So I think I think we all have been in that situation, and that same thing occurs in our research animals as well. So if you're studying cognition and learning, it's really important to make sure that we are decreasing stress. In the wild, stress can lead to abandonment of reproduction, and certainly in our captive breeding colonies, we see the same. And again, hormone alteration. If you're going to be studying hormones, this is very important to you. So I hope through all that, I've shown that this is important for the well-being of the animal. It's important for us to treat animals well, but it's also really imperative for the best research results. So it's a win-win when we do this correctly and when we think of it or think through this carefully. So I think a previous presenter also mentioned increased healing from from wounds and surgeries. That's another benefit of research if you're using a protocol that involves a research procedure. Again, if you're studying a vaccine, you would want to have an immunological response that's like the general population of birds. And again, cognition, learning, and are also affected. Okay, so the research environment certainly presents some limitations and some sort of framework where we have to work within in order to, when we're developing our enrichment programs. So things that would make research animals slightly different than other types of aviculture would be that sometimes our caging can't be quite as big because we need to have a larger number of animals in the study. And sometimes our cages are purposely smaller if we need to handle them on a regular basis, and the capture itself is a very big stressor for the birds. So being able to have access to the birds is one reason. So our space might be limited. We need our enrichment items to be very durable and reusable, or very cheap and disposable. Some of our research projects may not last for a long time. And so the balance of a novel enrichment item and the stress of something novel, we really need to think through the kind of cost benefits to those animals, and think of ways, not that we shouldn't do enrichment, but that might limit the kinds of enrichment that we can do. Cost is always a factor, not that it should be a huge barrier, but budgets are typically are always limited. There's some limit somewhere, even in a really well-resourced program. For most research programs, we have the expectation that we will be able to disinfect all the parts of the cage at a certain interval, and that we have validated that method is working. That might not be required of all settings, but that is something that most research institutions require some sort of validation of our disinfection. We don't want to introduce variability in our research. So we want to think through how we're doing enrichment in a way that doesn't introduce a variable that would cause our results to be not what we think they are. And of course, safety, as with any environment, we need to make sure it's safe for the birds. And sometimes that's a little bit complicated to assess because we may have a large number of birds and a limited amount of time to monitor those individual birds in a group. So we do have to be understanding that we need to be able to say that they're safe and that ideally that they're being utilized, but we might not have a ton of time to observe that. Some of the research settings, it's difficult for us to know if they're using their enrichment because, again, of that limited observation time. And resource guarding was something that's mentioned before, but we don't want to create a situation where there's limited resources and that enrichment is a source leads to aggression because there's not enough to it for every bird in the experiment or in that cage. Sorry for the delay there. Okay, so some of our studies may be infectious disease studies, and so biosafety may factor in. Both of the institutions that I came from do work on wildlife disease, and some of them are human pathogens. Some of them are pathogens that we absolutely would not ever want to escape the laboratory environment because it could harm our native wildlife or our domestic animals. And so biosafety is a consideration for a lot of these types of research facilities. What you're seeing there on the left is a biocontainment aviary. It's caging that's meant to contain a biosafety level 3 pathogen, and that's a pathogen that can be transmitted through the air. And so it has a feeder in the upper left. It's gasketed, so when it's closed, the air is entirely sealed and it's sucked out through a HEPA filtered ventilation system. And so that is really great for protecting the humans and other animals in the facility, the humans that work with those species, if it is an agent that can be transmitted to humans. But it really makes it difficult to enrich the lives of those animals, but not impossible. So we might need to be working around them, but it absolutely, this is not how we would house, just this picture is not how we would house birds in that setting. We would be adding enrichment devices, we would be adding disposable enrichment. And at some interval, you're going to be opening those cages to collect samples from the animals in most studies. And so we use those brief periods where the system is not enclosed in order to change in and out enrichment. So yes, it makes things difficult, but it's not a complete barrier. I think there was a question in the last presentation about disinfecting those wood materials. When we are doing infectious disease studies, we need to be really careful that we aren't moving infectious agents on those materials, and organic materials can be more difficult to disinfect. So sometimes we do put things like wooden perches in here, but when we are done with that, might stay there for a short study, and then we would autoclave it or incinerate it. And some things like wooden perches can handle a few rounds of autoclaving, but they probably won't make it through that many. So we often choose things that are disposable and cheap in order to enrich these environments. Cloth is another thing that's really difficult to disinfect, but I did want to add that one type of cloth we use is a surgical towel. So they're meant for surgeries. They are very durable for autoclaving and washing many times. And so we often use those when we are trying to create enrichment with cloth. So the kind of standard for disinfecting using institutional research cage washers is that most of them will go to 181 degrees C, and there will be some pressure involved. So it really beats up a lot of the reusable enrichment in devices that we have. And so if you're not going to be using that kind of high temperature disinfection, you would then rely on a chemical disinfectant, which you can for plastic items that would melt in an autoclave or in a high temp washer. But that is probably going to take more time to do to wash individual enrichment items. One reason that we might be housing animals in cages, even when there's not a biosafety, might be that we need to sample them on a very frequent basis. And so capturing a bird or in a large environment might actually be a very big stressor on them and potential for injury. So if we need to collect passive collection of urine or feces, we might have them in a cage like this that has part of it with a drop through wire cage in order to collect those samples. And we might have them in a cage where doesn't allow full flight for a short period of time in order to collect samples during a short study. There's some instances where we have a smaller environment, a cage or a smaller enclosure that is there because we need to be able to see the animal at all times to do video observations of them for data collection. So they need to stay in some space where we can see the details of their behavior, but so they're somewhat confined. So here's an example of a caging system that we used for a study where we had to collect daily samples from mallard ducks. But you can see we did, even though the cage certainly didn't come with many ways to add enrichment, we hung a bunch of kids' toys there and some mirrors. We reduced the amount of drop-through wire where the birds were on wire bars. And I do just want to add that when you have a confined space and you're adding enrichment, the balance effect is that you need to make sure that your enrichment is not taking up too much of their space. The standard for some species is to calculate the floor space, the area of the floor. And you might be better off to hang the enrichment rather than have it on the floor so the animal can still have as much space as possible. We also need to be really concerned about not introducing unmeasured variables. So all animals should be exposed to the same enrichment if that's possible. And the reason that, as I discussed before, is we don't want to be introducing variables that are not, that could alter the results. So giving treatment A different enrichment program than treatment B, it would really confound the results of that study. So as much as possible, we want to keep them equal across the treatment groups. Also, it's really important, or maybe important, to understand the experiences of the animals as they were developing, or their previous experiences with enrichment and caging systems. This picture is from the publication listed at the bottom, and what you're seeing is a visual representation of how the voltage-gated potassium channels in zebrafinch neurons are actually physically and chemically different if they're raised in a colony setting versus a pair-reared setting. So in that example, the history of an enriched life during neural development is important to know if you're understanding how the neurons are functioning and looking at a later point in the experiment. And also, we really want to be able to record enrichment, because if there is an unexpected effect, you will not be able to investigate it and find it if you haven't accurately recorded enrichment for each group. And then there's also some types of studies are the same group of animals are reused across time. And so if you, during the period where they're receiving treatment X, you would want to standardize not every single piece of enrichment, but more like give the same level of enrichment when that same group of animals is experiencing treatment Y. So again, standardizing across treatment groups. So all animal caretakers probably know this really well, but taking care of animals, especially a lot of animals in a zoo or a research setting or a rehab setting, is a long, busy, physically exhausting day. So there is some practical limitations to how much we can do when we have a research institution with many, many, many birds. I don't want money to be an excuse for not enriching the lives of our research animals, but it is a limitation that is just a real life limitation. And when we have, say, a lot of enrichment that needs to be disinfected manually with a chemical disinfectant and a manual scrubbing, that's going to really, really increase the time that an individual animal care technician needs to clean each individual cage. And so we need to, that is a limitation, in that we may not be able to just immediately hire more staff, even if we have the money. And all of that staff's time is money, which we may not, we may have a fixed budget at the beginning of the year. So we're trying to utilize things that are cheap and disposable or easily disinfected, because we may be doing this for a lot of cages. And also we want to think about short-term studies versus long-term studies. So the studies where the animals are going to stay in the same environment, maybe be used for repeated types of studies, they often we have the ability to develop enrichment that can stay in the cage and be reused longer. Also, the animals have more time to get used to that novelty. Whereas the short-term studies, we are a little bit more limited for both of those same reasons. The animals might not have that much time to adapt to the novel enrichment, and we might not have a lot of time to adjust our procedures and our staffing and our resources for that short-term study. So how do we meet the goal of encouraging species-specific behaviors? Well, you've already heard one framework that was very similar presented about how to divide and categorize enrichment, but we use this system more so that we can make sure that we are encouraging as many types of behaviors and experiences for the animal as possible. So the categories are meant to help broaden our the breadth of the kinds of enrichment that we're doing. And it's not actually that important, like which category each thing fits into. So don't get too hung up on how my categories are different than someone else's categories, or how each individual enrichment might fit actually 2 different categories. It's good that they overlap. But the categories that we use are physical or occupational, and those are things that encourage natural behaviors like foraging, nesting, and even mating displays or reproductive behaviors. Our social interactions, our sensory enrichment utilizes our physical senses, and our dietary enrichment would be different types of foods. And then environmental enrichment are changing the environment that they're experiencing to meet more complex. Okay, hopefully this video works. As you can hear, that's a lot of happy zebra finches. This is from our zebra finch colony at the National Wildlife Health Center. Zebra finches are social gregarious birds native to Australia. They perch and move through the environment in large groups, typically perching in trees together. Because of their size and their small feet, they're often choosing smaller branches that are highly mobile. They nest in groups, but within the group they have pairs and they defend their own space. Their diet is mostly seed-based diet. The males build a nest for the females and then sing and groom to attract them. So that's the wild context that we're trying to recreate here in this laboratory setting. And so you can see we have a variety of perches. We have nesting material with that shredded paper product in the middle. We have some natural leaves that they can use to manipulate and also to hide in. Other things, I'll show you a number of other examples. We have their seed located in multiple locations throughout the cage, both on the cage floor and in cups on the cage walls. And outside of the individual groups that they have in this cage, this particular example is a female only cage. Oh, I'm incorrect. That's a male cage. So it's a group of males, but they're housed in the context of a whole room full of zebra finches. So they still experience that large group setting, even though they're in a cage of 6 or 8 finches. Oh, I think I skipped a slide. Well, I thought I had a video here of some ducks, but if it comes across later, I'll describe it. So the first category is physical or occupational. So nesting and building nests is one of a very common behavior for many species of birds. So we almost always give them nesting material, even when they're not in a pair male-female group. Zebra finches love to find and play and manipulate with nesting material. And they may create nests, or they might just shred it. So what you can see in the left is actually a coin wrapper filled with shredded paper, and then it's clipped on with a little binder clip. So that's a super cheap disposable thing that the birds can use for nesting material. They can shred it, and because of how it's clipped on the cage, it's not super easy for them to get into that open side. They do have to fly around and use some of the other perches. I would also add here that the picture in the middle is actually 2 males in a nest that they've created together. So even though the natural behavior is for a male to create a nest to entice a female, when we had them in same-sex groups, the males used that nesting material and they made beautiful nests despite the fact that there was no female there. And they often stayed in the nest together. And there's evidence that it's the process that's enriching for them, not the product, in that they often made these beautiful nests that you can see on the right side, but then a few days later, they would completely dismantle it and start it over. So it seemed to be, for the birds, the thing that was rewarding was the process of making it, not the actual perfect nest. And as I said, even when they're not in, these birds were intended to be used for reproductive toxicology research. So we wanted them to continue to express their reproductive behaviors, even when they were not actively reproducing. And we found some creative ways to do that. So we also utilized fake eggs to allow them to incubate and have a nesting pair with a nest, and the female would lay eggs, and we would remove them and replace them with similar sized eggs that allowed them to continue to display those pair bonding behaviors and incubate their eggs and have all those natural eggs without continuously laying, which can have negative effects on calcium metabolism, and without producing more birds than we were going to use. So there's my video. So here's my happy duck video. So ducks naturally forage, waterfowl, many of them naturally forage in the water. And so whenever possible, we try to give them food in a pool or in a water, even in a cage setting, and it could be in a small little Tupperware container. We can provide their diet, or at least their enrichment foods, in water. That is really, they will strongly prefer to get things out of water even than out of dish. We can use floating greens, dried mealworms. Missouri actually makes a waterfowl diet that floats. And we also have social interactions in this picture. So these ducks were captive raised together. They have a pretty stable social structure, and they're housed in same-sex groups. If you are it was mentioned before regarding waterfowl, but if you are going to group house mixed males and female waterfowl together, be very cautious. Wild ducks can be very aggressive to each other, and resource guarding and mating are two of the things that really trigger those aggressive behaviors. So I absolutely always try and socially house ducks, because when they are not in large groups like this, they are very stressed. That part of their safety is being in groups, but that doesn't mean that all naturally occurring behaviors are something we want to recreate in our lab setting. And water access, as was also mentioned, is really important for prevention of bumblefoot. So I always give waterfowl water pools to float in. Okay. So I've already discussed a little bit about social enrichment. So it means allowing animals to interact with other compatible individuals to fulfill their social needs. What is their social need will depend on their age, their sex, their reproductive status, and what is the species norm. So the Office of Lab Animal Welfare wants us to always house species socially, except when they're approved for experimental reasons or veterinary reasons. Or individuals might be different than their species. So we need to consider individual compatibility. And despite these zebra finches being a gregarious, highly social species, in order to manage them in groups, we had to do a lot of regrouping. There was constant monitoring of who was picking at who and monitoring for aggression, and then regrouping them. Some of our technicians have an amazing ability to remember all of these negative interactions from the past and sort of intuit who might get along with each other the best. And so we had to spend a lot of time regrouping them in order to keep them happy and not aggressive. And in the upper right, you see a bird named Toby, who was a male zebra finch that we tried over and over and over again to find him a compatible cage mate, and we could not. And we just eventually came to the conclusion that Toby was happiest by himself, but in order to recreate a social group for him, we created a cardboard zebra finch. And Toby actually preferred to perch next to the cardboard zebra finch and would perch on it or next to it most frequently, and despite having lots of other space to perch. So maybe he just enjoyed a silent friend. And then the picture on the bottom right is another normal social group. It's a male and female paired with their clutch of fledglings. Here's some other examples of social housing. Again, that picture on the right is two males in a cup together that they've turned into a nest. That's not a nest box. That was their food dish. The ducklings on the left, we always house them in groups as well. And for some growing species, it's really important for them to have social learning. So for the ducklings, we see that they will follow when one individual starts exploring their environment and pecking at something, all the other ones in the group will follow and go investigate, is that a food source? And so they're actually learning from each other about their environment, what is food, what is not food. And as I mentioned, for ducks, having them in groups is also a predator protection strategy. So whenever we have them, it is ideal to have them in a bigger group. That helps them feel less stressed. So sensory enrichment refers to utilizing your senses of smell, taste, touch, and vision, hearing, and then I would also add vestibular or balance sense. One of the things that the zebra finches love to do is swing on swings, and many other birds do as well. And that's really engaging their sense of balance. Again, giving them perches that move engages that vestibular sense. The picture on the lower left is not a food dish. It's actually a water bath. So they love the tactile sense of the water, and they love to bathe. So that's, as soon as the human is out of the cage, the birds will come down to the water bath to bathe. We do want to tailor our sensory enrichment to the most developed senses of that species. So different types of raptors, for example, may hunt using visual cues or primarily olfactory cues. Most are a combination, but for example, some hawks may be really visual cues, whereas other raptors might be really cued in on scent. So we can use both of them, but maybe choose the one that's most developed in that sense. Whereas other birds are going to be using color and olfaction for foraging. And so this rosy-faced lovebird might be using both of those senses naturally for foraging. And so we want to stimulate them in the lab environment. Oh, there's our finch bathing. They do love the bath. Okay. Other senses, olfaction. So we often use birds. Another video of finches displaying them being on branches that are moving around the environment, and also the sound that they're displaying in a colony. So this is actually a YouTube video, and I would often play it for the birds while I was in the room. So you want to use especially if it's their own species call, you do want to be careful about not accidentally playing them a territorial or alarm call, but playing them the sounds that they might hear in their natural gregarious social environment is great for them. The sense of smell can be, we can use herbs that are very smelly, like cilantro, sage, other things that we can put those in their environment. For the things that are safe for them to eat, they can, like our zebra finches and ducks, I don't think any of them actually ate the cilantro, but they love to play with it and smell it. And then up in the upper right, there's a bunch of different natural sound things that are often marketed for sleeping or calming or meditation that have nature sounds. And so that's a really cheap and easy way to play some natural environmental sounds like rushing water, waves, or birds, or other types of nature sounds for our animals. So diet, oh, I'm almost at time. Oh, wow. There's also dietary or nutritional, which has been covered well by others. Just make sure that it is a balanced diet. There's the veterinarian aspect of making sure that our dietary enrichment is not too much of the diet. If it's millet alone is great, but it's not a complete diet. Our environment is changing the environment to make it more complex to mentally stimulate, and might be different textures of on the floor or on the substrate. It might be a mental stimulation of a puzzle. And then I just want to add novelty, especially for wild animals, but any animal, you can't have too much of a good thing. So you can have it be too scary, and that's going to be somewhat species specific, but there's also going to be some differences depending on individuals and what's their life experiences. If they're captive bred, wild caught, were they captive bred in the same setting or in an agricultural setting? So parrots tend to be really lovers of novelty. Juvenile animals tend to like novelty more. And but we do want to balance not creating too much stress. And then just the last few points, we want to document our enrichment. If you, again, to make sure that we can measure that potential variable. And we want to assess it. And I love to use camera traps to observe without being there in person. So we can take away the how much our presence might affect how they interact with the enrichment. And there's individual variation across a species. So what might not work for one duck could work for the next duck. These are all examples of things that sounded really good, but have failed on an individual group of birds. And that's it. Thank you very much. And I think I'm out of time, so I will have to hold for questions, I think.
Yes, ma'am. Thank you, Liz. Yeah, we're out of time, so we got to get on to the next presentation. However, I do appreciate your presentation. And if you have questions for Liz, you can ask her directly using the Whova app.